Thomas is professor of medicine. He's head of the Internal Research Institute in Strasbourg, and even more important, is a chair of the Hepatology University Hospital. So it brings together basic science and clinical science, which is actually the goal of this yellow webinar. The yellow webinar is where we can try together to link the two aspects. So I'm glad that and happy and grateful that Thomas rearranged his schedule to catch up with what we released 2 months ago. And the title of his presentation is Unraveling Liver Disease Biology and Discovery of Novel Therapeutics Using Single-Cell RNA-seq. So Thomas, the podium is yours. Thank you very much.
Thank you very much for the invitation, and thank you also very much for the kind introduction and talk we had before, Claudio. So today I would like to really show you recent advances in single-cell RNA-seq, and I'm sure you have read all the papers, and also you have starting to use, I think many of us now are using single-cell RNA-seq already as a routine in basic science and translational research. And here I just want to show you how basically we started and put together the first human liver cell atlas, and how we can use now single-cell RNA-seq to understand disease biology, but also to, at the end, unravel new disease mechanisms and new therapeutics. So today my talk will be about 40-45 minutes. I will start with concepts and methods of single-cell RNA-seq. To those of you who are not familiar with what technologies do we have, where are the pros and cons. I want to show you the Human Liver Cell Atlas, which serves for us and many all over the world as a reference for the study of liver disease and cancer. Then move to how can we use single-cell RNA-seq to understand liver disease biology, and then give you an example how we can evaluate new therapeutics using patient models and combine with single-cell RNA-seq, and then the conclusions and perspectives where I think the field will move in the future. And please feel free to ask any questions also in between. I think this is a very easygoing seminar. So I want to start basically very conceptual. And in medicine, we have been looking if we want to study the patient and this patient disease, we have really seen a revolution in the last couple of centuries, and then I think in the last decade. So let's start with an example of imaging. We have what we're still using is the inspection. Simply look at the patients, and then I see here a belly with ascites and representative for liver disease. Then basically radiology on the right side. You see here CT, and now we use MRI for here, for example, for the care of hepatocellular carcinoma. And similar in pathology, we had basically again the autopsy, necropsy here, an example from Rembrandt von Rijn in the 17th century, and then basically all the advantage in the histology. And in the recent decades, we had basically genomic bulk analysis and RNA-seq analysis. Now, one technology in the last 8, 9, 10 years, I believe, is really revolutionizing our understanding of disease biology and cancer, and this is single-cell RNA-seq. So this is here, again, very conceptual on the left side from a review we published in Journal of Hepatology less than 2 years ago, where you see a normal liver and a diseased liver composed of the individual cells. And now if we are able to isolate individual single cells and look at the transcriptomic state, we are able to understand the disease biology among several levels. And one is shown on the right side. This method allows us to look at the heterogeneity, very important for cancer, but also in liver disease, cell-cell interactions, disease-specific cell types. So we compare disease, and I will show you an example later on. And we are also able to identify rare cells, meaning new cells, which we have not known before, and understand their role in the pathogenesis. Furthermore, during special algorithms, and we worked with a group at the Max Planck who were really experts in that, in development, this also allows us to understand the development of cells in a given population or even in a given organ, and then deduct, for example, the origin and the phenotype of precursor cells to differentiated tissues. And I will show you also drug and target discovery and novel diagnostic and prognostic tools. So how can you do, what are the methods? How do you routinely perform single cell RNA-seq? And this is, I think, is a very important slide, because it's important if you, for example, have a specific questions or you have patient samples and you want to go now ahead with single cell RNA-seq, what method to use with which providers or which academic lab you want to work together. And there are basically two types of technologies which give different resolution and results, and also are different in terms of their expensive. So we have the array-based, where you basically, the cell is captured on plates. For example, we have been working with 384 well plates for the single cell liver atlas. And the other one is microdroplet, where you have droplets, and basically then you see these cells running through these droplets, and then are basically reverse transcribed, tagged, and analyzed. So the big difference is the array-based method, the called SmartSeq2 or seq2, is much more higher in the resolution. So it's really, you can go in depth, you cover most of the RNA species, but it's very, very expensive, and you also have a much lower yield, so you cannot sequence so many cells. The 10x has the advantage, you can sequence many, many cells. However, the resolution is lower, and if cells are fragile, so we observed this with the hepatocytes in particular, and that's still a challenge. Hepatocells and also cancer cells, they are basically fall apart in these microdroplet assays, whereas the array, if you basically use the protocol of using collagenase and digestions, you can adapt that and optimize that. So this is, and the other one is price. So the SmartSeq2 high resolution is expensive, whereas the 10x is quite affordable. And I think that when I started this, I think the price has gone down now, almost fivefold compared to, let's say, 5 years ago. So what we did then, it all started with a sabbatical I did at the Broad Institute of Harvard and MIT in Cambridge, Massachusetts. So where basically Aviv Regev at that time had already done single cell RNA-seq from several other organs. She's now head of research and development in Genentech. And so basically I realized that's something which we really need to apply for liver disease. And basically at that time we had really the ambition to start to do a single cell atlas of the human liver. So since it's impossible to have tissues from completely healthy individuals, we decided to use liver resections of diseased and non-diseased tissues from patients undergoing liver resections. So most of them for colorectal cancer, but also others. We then used flow cytometry to dissociate the cells into single cells, and then sorted the cells by flow cytometry in 384 well plates. Again, that's the array method, sequenced using these two protocols I show you here, and then did the bioinformatic analysis. And then we did data analysis and validation studies. So and this was a collaboration actually with the Max Planck Institute, Dominic Grün's group in Freiburg. So but all the patient work, everything related was done in Strasbourg. So so we at that time, we sequenced more than 10,000 cells from 9 different patients without liver disease. And so now the atlases are even bigger, but at that time, this was really huge already. And this allowed us really to get significant insights in the biology of the human liver. So this is a TSNE map. You have seen this now. You see this now in almost every journal in studying organ pathology. And what it shows basically is a T-distributed stochastic neighboring embedding, so where basically a high dimensional information is compressed in a 2D space. So basically we try to have the 3D space and a 2D space. Each dot represents a single cell based on the analysis, and the distance between the cells is a function of their transcriptional similarity. So it's by a method called stochastic neighboring embedding. And then marker genes, for example, albumin for the hepatocytes, enable identification of known and novel cell types. And you see already we have hepatocytes. We have actually an overrepresentation of the non-hepatic cells here. And this is simply, again, due to the fragility of the hepatocytes. And what we see here, we have the Kupfer cells. They are quite apart. The immune cells, B cells, endothelial cells, stellate cells, and very interesting, the EBCAM positive progenitor cells and the cholangiocytes, they cluster in the same cluster, and I will show you some more data on that. So now how can we apply that? And so the first things we did really, we asked the question, can we use this, can we apply this for simple liver biology? And here we started here with zonations, the notion you all have learned in your graduate or undergraduate studies or medical school. The liver is really a key example for a zonated organ. And what we basically could show in a combination of bioinformatic analysis and also then validation of immunostaining from the Human Protein Atlas, that basically zonation is not only the case for hepatocytes, but also for other liver resident cells. So here we have shown you an example for the endothelial cells and the NPCs. We see co-zonated genes and functions across hepatocytes and endothelial cells. And what was very interesting, that the comparison between the mouse and the human revealed only limited evolutionary conservation of zonation. So then we moved on to, let me see, now it's not moving here. Now it's moving. Yeah. So then we moved to better understand the question of progenitor cells. And this was, I think, one of the key findings, which at the end also got us into Nature to say, can we apply this this atlas now to learn something new, and something new which is likely important for understand the biology and the pathophysiology. And so here our colleagues at the Max Planck, they had developed a special algorithm called Fate ID, where they were able to understand the fate of different cells in a given population. And so using this technology, we identified a new cell population, which was characterized by EBCAM positive cells and had a TROP2 intermediate population. And this cell type was able to differentiate into both cholangiocytes and hepatocytes. So this made the discovery that they indeed, this is a question which had been discussed already for a very long time, that there are indeed bipotent progenitor cells which go then in one or the other direction. This is also interesting, and I'm not showing it today, but we have a paper in Science Translation Medicine in final review, where we looked at basically a new target called Claudin-1, and we actually show that these EBCAM positive cells, they play a key role also in these precursor cells in liver fibrosis, and can be targeted by an antibody. So that's also a finding not only for the biology, but also for the pathophysiology. And on the right side, you see the validation again by immunohistochemistry that these cells exist in the human liver in a patient. So then the next was cancer. And cancer, there's been now a huge progress. So actually, we were the first at that time who basically looked into basically were able to compare healthy and cancer cells actually from the same patients, basically the adjacent tissue and the cancer cells, and study get first functional insights about cancer biology in HCC. So these were 1,000 cells from 3 HCC. We had alcoholic liver disease at that time. The goal was not to do this comprehensively, but simply show proof of principle, proof of concept. And when we then, first of all, what you see here, the first thing you notice, that you only have actually a small number of cancer cells. And here in this study, the majority of these cells we isolated from the cancer were NPCs. You see, we matched them simply to the human liver cell atlas in gray, and then in red, the cancer cells. And what you see here, basically how these cells co-localize, and only some cancer cells. And this is again due to one, of course, this could be cancers which have a large NPC fraction, but most likely here, again, it's the isolation method. So if we then look at the phenotype, and then simply we can zoom in how basically the NPCs different in the tumor compared to the adjacent tissue. And here, for example, I show you 3 examples. We looked at the endothelial cells. They were enriched for extracellular matrix and tyrosine kinase expression. So again, showing a contribution also to cancer HCC fibrosis. The NK cells showed again an upregulation and a downregulation of metabolism, and the macrophages showed a downregulation for antigen processing and presentation, showing again, so we looked at CAFs and tumor-associated macrophages. And again, this already gives, I think, very new insights into the cancer biology. This was then basically, was then reproduced and then extended. There are now several single-cell RNA-seq studies published, and I don't have the time to go through all of them. This is one I think is a very good one, nice one, has basically HCC and also some cholangiocellular carcinoma patients. It's from Tim Greten and Dr. Wang from the NIH group, and they basically they looked into a patient cohort, and these are all publicly available. And we're actually using these data also for example, now to understand certain targets. They showed basically that functional clonality is a prognostic surrogate of tumor cell fate. And they showed basically the evolution of the tumor cells is linked to, again, to the immune system and pre-exhausted cells, what has been postulated and is being studied very heavily. And they found osteopontin SPP1 was tightly associated with tumor cell evolution and microenvironmental reprogramming, which is also, I think, gives, again, substance to what is known already about osteopontin in liver disease biology. And also what they found that patients with tumors with a higher cluster number had a much shorter survival. So this shows you again already that we in the future, it's still early because we need liver tissue for that. We need a setup for doing this, but I think there's also a potential to use this method at some point, integrate that into the clinical care, for example, by stratifying patients for HCC and into potential responder, non-responders, or for the targeted therapy. Now, another application is virus-induced cancer, and as you know, we have been working for the last 20 years on virus-induced liver disease and cancer. And what we looked here, basically very simple, what about hepatitis B virus interactions? And we found actually that the virus was compartmentalized. You see this in panel B. There's clusters with higher and lower HBV RNA, and which is also has been known already for hepatitis C virus. And what you see here, we can actually now also understand how the integrated HBV here, because the RNA, we can basically map that to the integrated HBV. And what we see here is that basically in which fragments HBV RNA is transcribed into host cell RNA or DNA, then to understand HBV new integration sites. So this basically is one example out of this. So again, it shows you that and this is now heavily used in the field. I'm an editor at the Journal of Hepatology, and basically we're receiving now many papers also now studying in detail in patients, for example, the role of single-cell RNA to understand, for example, mechanisms of viral clearance and persistence. So one more last example for the disease biology before I move to drug discovery. This is, I believe, is a fantastic paper from our colleagues from Neil Henderson in Edinburgh, Ramachandran et al. And they basically, they went to the next level. They compared healthy and cirrhotic liver. So this is here, you see this on the right side. So they had 5 healthy patients from 5 patients, 5 from cirrhotic patients. Also, they had the blood. And here in this case, they used the 10x method. So meaning they had mostly NPCs and just very, very few hepatocytes. So this atlas is therefore focused really on the non-parenchymal cells. But nevertheless, this gives very, very, very nice informations and had allowed to discover new cell types. So then they analyzed, they called it the fibrotic niche of the human liver, basically meaning what are the key differentiators, and identified novel cell types. And one of them, which are now have been also studied by others and several follow-up papers, are the TREM2 CD9 macrophages. So how did they do this? I don't want to go into detail. This is all you can simply do by bioinformatic analysis. So most of the data are really simply bioinformatic analysis, and then they did validation studies. So here, this TREM2 encodes for an innate immune scavenger receptor, is implicated in phagocytosis and clearance of apoptotic cells. And they are basically using these data. They showed that these macrophages interacts with scar-associated endothelial cells and mesenchymal cells in the fibrotic niche. So this is shown here. And again, these data really, this is from our review we wrote then with Neil Henderson in Journal of Hepatology. And this is what I think is really fascinating, that this model now of NPC interaction is derived from really a simple bioinformatic analysis from patients and with some validation studies. So again, here they basically show their model for the interaction of these scar-associated macrophage with the mesenchymal cells and the endothelial cells and the receptors involved, with the most relevant molecules belonging to Notch, PDGF, VEGF, TGF-beta, and TNF families, which I think you are all familiar. So we know as players already for a long time, but I think this paper gives us a new understanding in terms of how the different cell types interact and which cell types are existing in the fibrotic niche. So I want to summarize this first part of my talk, and what I think, what really I think the last 5 years have shown that single-cell RNA-seq has paved the way for the discovery of previously unknown cell types. I've shown you, for example, the macrophage in the fibrotic niche, and subtypes. I've shown you also the TROP2 intermediate phenotype progenitor cells in normal and diseased liver. The study of the phenotype and the functional role of these is transforming our knowledge, in particular of the liver microenvironment. As I told you, first, because it's much easier. It's easier to get the information on these cells due to a technology. But second, also we are learning now from many different types of cancers in particular, but also fibrosis, that the NPCs play a key role in disease outcome and now are being tried to be targeted by different therapies. I've shown you the cell-cell communication, and what I will show you now is an example which we did, and we have several others, how we can use single-cell RNA-seq combined with functional study to discover novel drugs for NASH, fibrosis, and also cancer. So now I just wanted to, do you have any questions here? If not, then I think
we will make the question at the end of your.
We will do the questions at the end. Yeah, perfect. So now, let's move to one of the key challenges we face now in hepatology, and this is liver fibrosis and HCC. So I just want to remind you again that this is really a major public health burden with limited or absent therapeutic options. We have, first of all, the challenge of the rising prevalence. Chronic liver disease affects up to 800 million people worldwide, about 2 million deaths per year. The fibrosis global prevalence is between 1.5 and 6%, growing rapidly. There are 3 million patients with advanced fibrosis in the United States alone. Why is this important? Liver disease-related mortality risk is 42-fold higher in patients with and without fibrosis. You all know more than 80% of the HCCs arise on the fibrotic liver. And at this point, we don't have any approved anti-fibrotic therapy, and still there's a very limited efficacy of the compounds in clinical development. There's clearly, I would say, at least that's how I see that, not yet a breakthrough of a compound which we think we can solve this problem easily. So this is here again. I like this cartoon very much because it's very simple, but illustrates the fibrotic cliff which we challenge. And fibrosis is caused by alcohol, metabolic disorders, NASH, HCV, and HBV. And then that's basically what we also learned, that actually the clinical cause and also many of the mechanisms are shared between these etiologies. At the end, all these etiologies result in F3, F4, advanced fibrosis, ultimately into cirrhosis with a point of no return. We know this, for example, for HCV clearance. If you cure patients too late in cirrhosis, basically patients then still progress with decompensation, HCC, liver failure, and death. And there is basically, we have learned now in the last really decade, it's possible to partially reverse early stage fibrosis if you remove the etiological driver. This has been nicely shown for diet bariatric surgery, where especially with steatosis, control and cure a viral infection. If you cure not too late, or you withdraw alcohol, then we have the possibility of a reversal of fibrosis. However, as I just mentioned before, there seems to be a point of no return in cirrhosis, basically where then, for example, through epigenetic changes, we have shown this by HCV, if let's say the injury has left so many footprints in the liver and the overall organ architecture is already destroyed, that there's a point of no return where fibrosis is not anymore relevant. And second, it's even if you remove if you, let's say, a patient with NAFLD, you basically do everything, diet, exercise, the reversal is only partial. Now this here I think nicely illustrates the HCV case, which I just mentioned, two landmark studies from El-Serraq and from Kanwal. They showed basically that basically this is a large retrospective cohort of more than 22,000 patients treated with DAA in the VA administration for close to 40% of cirrhosis. They showed that that SVR was associated with a considerable reduction in the risk of HCC. However, in the patients with SVR, the absolute risk of HCC remained high in patients with established cirrhosis. You see this here on the left side. And then on the right side, this study from Strasbourg et al. They showed basically if you have then a cirrhosis, child BC cirrhosis, basically the treatment, the cure comes through late. Clinically event-free survival was not improved in this study. So this again shows the need for novel antifibrotic therapy. So what do we know about fibrosis? So these are some concepts. It's just from a review from Natasha Rullen from my lab, where basically we see a chronic liver disease and the hepatocyte injury drives liver fibrosis. So the injured hepatocytes is basically the key driver, is on top of the hierarchy of the fibrosis. And then we have stellate cells and macrophages, which play a key role. They produce an extracellular matrix. And then we have chronic fibrosis with this pro-carcinogenic milieu and with the here illustrated complex cell circuits mediating the pathogenesis of fibrosis. So there's, and before I will show you how we can use single-cell RNA-seq, I just want to show you the challenge we face at the moment. In the last decade, really a huge number of targets to impair progression of fibrosis has been studied. And here are some examples. This is from a review from Brenner, and showing targeting the injury by itself, the inflammation, the activation of the stellate cells, and also try to do fibrosis regression by inducing apoptosis. But despite all these major efforts and billions of resources, so basically investors always say NASH plus cash is ash, so meaning if you invest in NASH with a lot of cash, at the end nothing remains. So very frustrating. And this is shown here that we have been, these are drugs beyond phase 2. So there's some new drugs of course now on the basically in development. But you see here, so far there's no approved drug, and the results have been largely disappointing. There's still a possibility for approval of obeticholic acid, but if you look at the overall numbers, the improvement is really incremental. There are now some, there is a company called Akero. They have just presented some interesting phase 2b results with an FGF targeting agent. So of course there is hope, but so far we have seen that often in the phase 3, then these results could not be reproduced or were not strong enough, warranting FDA approval. So really a big unmet medical need. So why is it so difficult? Why don't we, after all this research and these billions of dollars, why don't we have a drug which works in fibrosis? One is the complex cell circuits mediating the pathogenesis. So I showed you this slide before. It's not like a single pathway which you can easily target and then you have the solution. It's really multiple cell types, including the liver NPCs. We still often don't know what is driving and bystander. So we know, of course, the hepatocytes are likely at the beginning, but then we have stellate cells, macrophages, which cell type is basically playing really the key role. The other big problem, which I think is a big reason for failure, is that the models we have is not good. So the current animal models, for example, these chemical models, CCL4, TAA models, they often only partially recapitulate the human disease. So mice and humans are very different, in particular with fibrosis. And just the compounds I just showed you, they are fantastic. You can really dramatically improve fibrosis in mice, but at the end, in humans, the effect is marginal. So we thought that we need new models recapitulating key circuits of patients' disease, and which are very simple, fast, and where we can understand new mechanisms and to uncover new drugs. So, and here this is actually come from a work I met when I was at abroad. I met Jujin Hoshida. Hoshida, many of you know probably, he was at that time a fellow at abroad, and he discovered in the lab of Todd Golub a prognostic liver signature. So this is a signature which is expressed in the tissue of patients with advanced fibrosis and cirrhosis. And this clinical prognostic liver signature robustly predicts survival, fibrotic liver disease progression, and cancer risk in all major etiologies. So this was his New England Journal paper in 2008. And this prognostic liver signature comprises 73 poor prognosis genes. You see the pathways here on the right side, and more than 100 good prognosis genes. So you see that genes, if they're upregulated, poor prognosis is TNF-alpha, NF-kappa B, the usual suspects, TGF-beta, interferon, and the good prognosis genes are genes metabolism. So if the metabolism is active and preserved, the patients will remain stable, not develop decompensation and HCC. So this signature can really very nicely in clinical studies. He has published several follow-up papers on that in gastroenterology, in Cancer Cell, and now also he has just this year a subset he has published in Science Translational Medicine and Gastroenterology that you can basically use a subset of the signatures for a blood-based circulating biomarker in patients and validate the signature again. So I believe it's really a fantastic signature. Now, how can we use this signature in research? Now, the challenge is in patients that patients develop this signature takes about 20 years to develop. So this is not something you can easily model. And we basically then, we asked the question, can we model this in a cell culture system to then identify the pathways and the drugs which modulate the signature to then in the second step to develop novel targets and compounds. I will show you one approach and one target as an example, and we have done many more. So I think the originality lies here. It's a multi-target approach, 186 genes covering the key cell circuits in contrast to classical drug discovery where you look at one gene or one cell type to find new drugs and targets. So how can we induce this prognostic liver signature? And there are different ways. The most easiest is using hepatitis C virus, and we have a co-culture system. We started first with simple HUH7 cells, LX2 cells, and TMP1 cells, so cell lines, and we then transformed this in primary cells. We'll show you in a minute. And so if we infect in this co-culture models, the cells, we can induce this poor prognostic liver signature in a matter of 14 days. And we then transformed this system to a patient-derived model because from cell lines, we are actually learning also from what is known, what we have learned in the past in terms of the key thing is to use primary cells and human cells. And here, basically, we developed a model system, Emily Crochet, together with our liver surgeons, where we create spheroids from patients with liver disease. So we dissociate these cells and then put them together on ultra-low attachment plates with growth factors. And then on the right side, you see here the patient spheroids. And the beauty of these spheroids is that these cells, they not only have hepatocytes, but they have really all the non-parenchymal cells. Like, let's see, you see here CD31 endothelial cells, Kupffer cells, stellate cells, and they also have immune cells like CD8 and CD8 for T cells. So this actually, the system, just to show you, is very specific for liver disease. So HCV, HBV, but not dengue virus induces the signature. Alcohol and free fatty acid induce this liver signature, but for example, not another stress, something like low glucose. It's very specific for the liver etiology inducing agents. And what then Jujin showed by looking, basically comparing with patient, this signatures, the changes we find in the cell culture are really very similar compared to what we find in patients. So showing that this model is really a clinically applicable and translatable model. So then we used the method. We did first an in silico screen. So basically this was with the CMAP and links library. Assessed 20,000 compounds which are available and checked their effect in databases on this prognostic liver signature. From this screen, we found 25 candidate bioactive compounds. So when we were simply actually redoing the screen at the moment. And what we found at that time, this was 2016, we found 25 candidate bioactive compounds which were active in that model system. And then we went for functional assessment in our cell-based system and selected the compounds based on the reversal of the PLS and then characterized the compounds. So this here on the right side, you see the drug discovery model. And what we found is the compounds which scored highest in this model were captopril and nizatidine. Captopril, you know, an ACE inhibitor, which is already, you know, the ACE inhibitors has been shown to work nicely on fibrosis, but they are basically not really tolerable because of lowering the blood pressure, but this is a very nice proof of concept. Then you see a large series of other compounds, pioglitazone, resveratrol in red wine, for example. But one of the compounds having the highest score was nizatidine. So this was very interesting for us. And because nizatidine is an histamine receptor 2 blocker, it's a very safe compound, has been used for decades in patients for treatment of gastric and duodenal ulcers. And so we decided to follow up on this to understand how nizatidine works and test nizatidine in proof of concept animal models for its potential use in patients. So first of all, we looked into animal models, and you see here, these were obtained from Brian Fuchs at Mass General Cancer Center, Harvard Medical School, who is now in Ferring. So we used basically two models. One is a rat model, the other one is a mouse model. We decided to use choline-deficient high-fat diet because we think it's more physiological. And what we induce here, advanced fibrosis or even cirrhosis, like in the rats. And then we intervened following the establishment of fibrosis, week 8 or week 12, we treated the animals with nizatidine and really found a striking effect. We have a very dramatic reduction of the collagen proportional area, alpha SMA, hydroxyproline, and TIM1, collagen 1. And on the right side, you see also the number of tumor nodules is markedly decreasing. So nizatidine markedly and significantly inhibits fibrotic liver disease progression and HCC development in 2 state-of-the-art rodent models. So this actually, we have published this in Nature Communications last year and a follow-up study in JCI on captopril this year. So here, the details again from the rat model, and I just wanted to show you again. If you look at I think particularly on the gross pathology, the vehicle-controlled and the nizatidine, you see really a dramatic reduction of the tumor nodules seen here also in the quantification. So again, ultimately what we want to really achieve is prevention of HCC development and decompensation, which we do here. So now, how can we understand the mechanism? So this was a new target. We didn't know, no idea how does that work, and how can we understand it? And here, actually, we quickly applied our human single cell atlas to understand the mechanism of action. This is an example I think I want to show you today, how you can use that for your studies as well. So first of all, very simple, and that's the first thing. If you talk to an industry or an investor, they want to know, where's your target expressed? And this is shown here. On this slide, we looked simply in our atlas, and what we found, the histamine receptor was actually highest expressed, not in hepatocyte, but it was expressed in macrophages. So this already gave us a first clue about the cell type we have to look at. So it's not the hepatocytes, but it's really the macrophage. So then we zoomed in on the macrophages. So again here, this is this TSNE map, and high expression is in red and in yellow. And we wanted to know which subtype of the macrophage is where is histamine receptor expressed. And then we looked at really at the co-expression of different markers for macrophages. As you know, there's a wide array of macrophages, the pro-inflammatory macrophages, immunoregulatory macrophages. And what we found actually that histamine receptor 2 was highly expressed in the pro-inflammatory liver macrophages, CLEC5 A high and MACO low macrophages. So now is the question, are the liver macrophages really the nizatidine target? How can you prove this? And so then Emily, she did a fantastic job. She took liver tissues, again, from patients either diseased or healthy, then dissociated these liver cells, then I put them in cell culture again, and then performed single-cell RNA-seq. And this allowed us to understand if now in an intervention study, when we add the nizatidine, what is the effect of nizatidine? So again, we take the tissue, we generate spheroids, we treat them with nizatidine, and then we perform single-cell RNA-seq. And this is shown here. Then we can basically look at each individual cells, which cells has responded or reacted to nizatidine, and what happens exactly on the cellular level. So it shows us exactly when the compound hits its target, and then at the same time, what does the compound do in the cell type? So what we show here, again, in these analysis, so these are basically, we have TSNE map, and then here on the right side, we have basically bar graphs showing that the nizatidine treatment resulted in a marked shift within the liver transcriptome, within the transcriptome of the liver macrophages. Again, we could confirm the macrophages which had a change in the transcription profile were the same macrophages where the receptor was highly expressed. Nizatidine also decreased pro-inflammatory, pro-fibrogenic, and pro-carcinogenic cytokine expression. You see this here, if you look on the right side, I'm not sure whether you can see my cursor, you can see on the right side in the bar graphs, each bar is a single cell. And then we can see the gene, and then we see, for example, here that how nizatidine up or down regulates the cell. So blue means in this case it down regulates, and red it up regulates. So you see here, we see these CCL2, CCL5, the interleukins, MMP7, 9, all pro-fibrogenic or inflammation markers, they go down. And then, for example, the genes for antigen presentation, the HLA genes, they go up relative to control-treated cells. So in summary, nizatidine induces a shift from pro-inflammatory, pro-fibrogenic phenotype to an atypical immunoregulatory macrophage profile. And this is, we believe, plays an important role in the amelioration of fibrosis and HCC prevention, not only likely for nizatidine, but in general. So here then we went back to our animal models. Brian Fuchs, Matthias Heikenwälder actually did the immunohistochemistry. And what we see actually, indeed, if we treat these animals with nizatidine, we exactly see this, what happens in the spheroids, that we have less macrophages, the F480, and we also have less, we have change in the T cells. Actually, in this case, we recruit and increase the CD8 T cells. We think that we have then a better tumor surveillance in these animals, which could mediate the cancer protective effect. So in summary, and these are now my last slides, I have shown you how single-cell RNA-seq could unravel a novel target and mechanism of action in fibrosis and liver disease. We identified the histamine 2 receptor positive macrophages as a therapeutic target for the treatment of liver fibrosis and HCC prevention. You see here the macrophage, which from our and other data is involved in the activation, recruitment, inflammation, proliferation, and inhibition. And if we now block this activation and the proliferation, we have an improvement of liver fibrosis and cancer prevention in our 2 animal models. We actually looked at also in patients of the expression and basically confirmed that basically the histamine receptor expression correlates with disease progression. So in summary, I want to close now. This is my last slide. I want to show you how can we use now these novel technologies for drug discovery. I've shown you here an example, a drug discovery platform, transcriptome-based in silico drug screening using this prognostic liver signature, allowed us to discover novel compounds with antifibrotic, anti-inflammatory, and HCC preventive properties. We have these perturbation studies then with RNA-seq, single-cell RNA-seq, and rebels to the mechanism of action of this compound. And we are currently working also, I created a startup company, Alentis Therapeutics, is now applying this CPLS system combined with single-cell RNA-seq as a simple platform for liver disease drug and target discovery, which is, we all agree, is a key unmet medical need worldwide. So with this slide, I would like to close and acknowledge. So really a big thank you to Emily Crochet, Antonio Saviano, and Natasha Roen, who did a lot of the work here shown, and the whole team at the University of Strasbourg, our collaborators, Judin Hoshida, Dominique Grün, Matthias Heikenwälder, Ave Fregev, Brian Fuchs and College and Harvard, and also our funders, which made these studies possible. And you, I thank you very much for your attention. I'm looking forward to exciting questions. Thank you.
Well, thank you very much for this excellent presentation. You can actually return to the screen so we can have a face-to-face discussion. Thank you very much, Thomas. This was a wonderful and inspiring presentation. And I want to start the question time. Is anyone with burning questions in his or her mind? If not, let's start to break the ice. I mean, I found your data very, very exciting.
Thanks.
But I have a question. I mean, I think there's no question that the degree of fibrosis is making the difference in the clinical outcome of it.
Yeah.
If you have fibrosis, so the expectancy of the rest of your life is shorter, and the clinical course is worse. But once you have cancer, probably it's good to reduce the possibility to increase the fibrosis, but probably it's too late. I mean, you can modulate. The question I have, shall we try to understand what's going on in a still reversible phase or curable phase? And I think one of the model may be the translation from NAFL to NASH or MAFL to MASH, because this is where probably we can have the best option to stop or to reverse the outcome. Do you have any data on cells collected from patients with NAFL or patients with NASH, in order to see whether your histamine drug may be one target to use to do it?
No, I think that's an excellent question. So first of all, I want to thank you for your kind words, and I agree with you. I think let's say if we want to work in a preventive approach for a very large number of patients, and I think then basically the early stage fibrosis would be the ideal target population. So that's basically also what many of the companies are trying. Of course, this would need to be a compound which also with a limited cost, but since it we would need really millions of patients, and I agree. The challenge is a little bit that we often we don't have any biological materials from these patients, so we have less ways to understand basically the progression. Here, I think there's the value of metabolic drugs is very important. Let's say also companies like Novo, they're very active in that. Novo Nordisk, basically also to have drugs which combine at the same time the underlying disease, improve the metabolism, and then in the second time basically also improve the fibrosis. I agree. I think this is one focus area, which definitely it has to be a key, and which is also the key. And the second, I think, as we did, and is basically to go into the advanced fibrosis where we are already late. I agree, but let's say if we are still before the cancer and we have the right drug and we can improve the microenvironment, I think we can bring down the cancer incidence.
No, I fully agree with you, but I mean, if I may still have a sort of additional question or comment. I mean, with all the limitations and all the caveats of comparing in vivo models with humans, because a mouse is not a man and rat is not a man, and most probably a mouse is not a rat and the rat is not a mouse. With all this limitation, I think that one possibility is to use models where we can modulate the degree of inflammation and fibrosis in
I agree.
young mice treated with some, not with chemicals, but with the nutritional factors. I think this can provide us a hint to see whether then we can translate to humans. Probably the answer is no, but I mean, I think that if the answer is yes, then we can have a major impact on the natural course of something which is really booming.
Completely agree. Yeah. And let me just share my screen one more time. I think this is it's actually what was in the annex of my presentation. So here we also looked, it's also in the Nature Comp paper, and the reviewers had asked for that. So basically we looked in different patient cohorts. So we looked into an HCV cohort, infected versus non-infected, where many of the patients actually were early stage fibrosis. We have an obesity cohort with basically obesity and NASH. So these healthy obesity, of course, they also have steatosis, but you see here also a nice difference. We have an HBV-HCC cohort, so this is of course late stage, and we have the Hushida New England Journal cohorts, which again were advanced fibrosis. So we see also an effect we have to go into the details. There are less data available for these early stage cohorts because patients are not biopsied routinely, but that's something we want to look at. So we thought this is you see there's basically pharma has all these compounds and we all participated, but we propose nizatidine as a very low cost, very simple approach. Nizatidine is safe, it's generic, it doesn't cost much to, and that's something we aim to do in a clinical trial, for example.
No side effect, actually.
Yeah, I just go back, stop sharing.
Still, I mean, there's a lot of overlapping. So I think we need a sort of a cooperative study to understand in large numbers in a short time.
You're right. No.
Okay, I stop here, so I don't.
Well, but thank you. I completely agree with you. I think that's the best approach is intervene early if we can with a. It has to be a drug which is really widely accepted because there's close to 1 billion of patients with steatosis. So if you that's a good point. Thanks. Yeah.
There's any question?
Don't be shy.
Don't be shy. I mean, there's any other question?
Technical question or
I have a question, Prof.
Okay, Alan, go ahead.
Thank you for that wonderful presentation. I'm just curious. So we have two types of RNA-seq, the bulk and then the single cell. So my question is, what situation is suitable for single cell or for bulk?
Yeah.
So
I think that's a very good question. I think the RNA-seq gives you really a very nice overview on basically your pathways and target. For example, if you have a certain target in your research, you want to understand it. You look at RNA-seq already. There you get information where it's at what stage of the disease expressed. Is it high or low? And then you can also deduct pathways already. If you with bioinformatic analysis associated pathways, but with GSEA, for example, you can do this. At the moment, if you want to know which are the players which are generating your protein expression, if you want to know which is the cell where the protein expression is high, then basically you need single cell RNA-seq. Then it can tell you this is the cell, for example, as I showed you. Here's the macrophage. Is it the hepatocyte? It tells you because you will see surprises. Sometimes even you don't see the upregulation in the bulk, or it's just very little. Because, but if you go into a single cell RNA-seq, for example, you see in the stellate cells, it pops up. It's really different. So this is this helps you the cell type, and then it helps you also to look at the cell communication to see are there cells basically what other cells and which cells could react to your target in terms of the downstream pathways. So the cell type, the cell communication. And also with spatial transcriptomics, as you know, I did not talk too much about it today. Basically, you take a tissue, and then basically you do single cell RNA-seq on a given tissue with keeping the spatial organization of the tissue. Then you can see to which cells your target cell talks to. You can really see in the tissue where is the crosstalk. So I think it's very. And the other question, of course, is time and money. If you want to do something quick and fast, I mean, you know, like an RNA-seq, you can get it for about 300 euros. A nice RNA-seq, whereas let's say a more elaborated single cell RNA-seq analysis, like it's about for let's say 3, 4, 5 samples, about 10,000 euros. So it's much more expensive. Meaning the whole, let's say 10,000 cells or so. It's we actually at that time just the single cell RNA-seq for this human cell liver cell was about 150K. So just to give you an example about the costs. So that's always much more costly. But prices go down, and I believe this technology will be a mainstream technology in a couple of years. So I think the liver is still, it's easy. That's why there's a lot of publications on immunology, blood, because it's easy if simply you take the blood, you run it through, you isolate the cells. The liver is still tricky because of the hepatocytes. They're so difficult to get them in a good condition for the sequencing.
But they are not the key players. They are not the key players. the hepatocytes.
You're right. Also, this could be on this side, but we still don't know. I think they're at the very.
Yeah,
I
but-
still believe on top of the cascade. They are you
Exactly.
have the injury, and then it all goes down.
But looks, they are more victims than the players, than the
It's
killers.
possible. Yeah, you agree.
The macrophages are
Yeah.
the big guys, in my opinion.
Yeah.
Okay, any other question? Well, if not, thank you again